Hi, today we are going to talk about how to exploring proteins and proteins. So as we know, proteins play crucial roles in nearly all biological processes, like in catalysis, signal transmission, and structure support. So a major goal of our course is to determine how amino acid sequences specify the conformations and hence the function of proteins. The purification of proteins is an essential first step in understanding their function. So how do we recognize the protein that we are looking for? Hence, the first step in these studies is the purification of protein of our interest. Proteins must be released from the cell to be purified. So first, cells are disrupted in a homogenizer, and the resulting mixture we call the homogeneous. Then the homogeneous is centrifuged in a step-by-step fashion of increasing centrifugal force. And the denser material will form a pellet at a lower concentration of force, and then with the less dense material. So the isolated fractions can be used for further purification. Proteins can be separated from one another on the basis of solubility, size, charge, and binding ability. First, we're going to introduce dialysis. So proteins can be separated from small molecules such as salt by dialysis through a semi-permeable membrane, such as a cellulose membrane with pores. Molecules having dimensions significantly greater than the pore diameter were retained inside the dialysis bag. So the small molecules and RNAs capable of passing through the pores of the membrane diffuse down their concentration gradients and emerge in the solution outside the bag. And then second is gel filtration chromatography. More discriminating separations on the basis of size can be achieved by the technique of gel filtration chromatography, also known as molecular exclusion chromatography. The sample is applied on the top of a column consists of pore beads made of insoluble but highly hydrated polymer, such as dextrin or agarose, which are carbohydrates, or polyacrylamides. Like self-dex, self-arose, and biogel are commonly used commercial preparations of these beads, which are typically 100 millimeter in diameter. So small molecules can enter these beads, but large ones cannot. So the result is that small molecules are distributed in the aqueous solution, both inside the beads and between them, whereas large molecules are located only in the solution between the beads. Large molecules flow more rapidly through this column and emerge first because a smaller molecule is accessible to them. To obtain a protein of high purity, one chromatography step is usually not sufficient because other proteins in the crude mixture will likely co-elute with the desired material. Additional purity can be achieved by performing sequential separations that are based on distinct molecular properties. For example, in addition to size, proteins can be separated on the basis of their net charge by ion exchange chromatography. If a protein has a net positive charge at pH 7, it will usually bind to a column of beads containing carboxylate groups, whereas a negatively charged protein will not. Affinity chromatography is another powerful means of purifying proteins that is highly selective for the protein of interest. This technique takes advantage of the high affinity of many proteins for specific chemical groups. For example, the plant protein concavalin A is a carboxylate binding protein, or lectin, that has affinity for glucose. when a crude extract is passed through a column of beads containing covalently attached glucose residues, concanavaline A binds to the beads, whereas most other proteins do not. So the bound concanavaline A can then be released from the column by adding a concentrated solution of glucose. The glucose in solution displaces the column-attached glucose residues from binding sites on concanavaline A. A technique called high-performance liquid chromatography, short for HPLC, is an advanced version of the column technique already discussed. The column materials are much more finely divided and as a consequence possess more interaction size and thus greater resolving power. In the sample HPLC elution profile shows in this picture, proteins are detected by setting the detectors to 220 nanometer, and that's the characteristic absorbance wavelength of a peptide bond. And in a short span of 10 minutes, a number of sharp peaks representing individual proteins can be readily identified. A molecule with a net charge will move in an electric field. This phenomenon, termed electrophoresis, offers a powerful means of separating proteins and other macromolecules. Electrophoretic separations are nearly always carried out in porous gels or on solid supports such as even paper, because the gel serves as a molecule sheath that enhances the separation. Molecules that are small compared to the pores in the gel readily move through the gel, whereas molecules much larger than the pore are almost immobile. Electrophoresis is performed in a thin vertical slab of polyacrylamide gel. A microliter pipette is used to place solutions of protein like in the well of the slab, and a cover is then placed over the gel chamber and the voltage is applied. The negatively charged SDS protein complex migrate in the direction of the anode at the bottom of the gel. And the sheathing action of a porous polyacrylamide gel separates proteins according to size, with the smallest ones moving most rapidly. Mixtures of proteins from cellular extracts subjected to electrophoresis on the SDS page gel can be visualized by staining with C blue. The first line contains a mixture of protein of known molecule weights, which can be used to estimate the size of the bands in the samples. So small proteins move rapidly through the gel, whereas large proteins stay at the top near the point of application of the mixture. The mobility of most polypeptide chains under these conditions is linearly proportional to the logarithm of their mass. The electrophoretic mobility of many proteins in SDS page gel is inversely proportional to the logarithm of their mass, and proteins can also be separated electrophoretically on the basis of their relative contents of acidic and basic residues. The isoelectric point, short for Pi, of a protein is the pH at which its net charge is 0. At this pH, its electrophoretic mobility is 0. Suppose that a mixture of proteins undergo electrophoresis in its pH gradient in a gel in the absence of SDS. So each protein will move until it reaches the position in the gel at which the pH is equal to the Pi of the protein. This method of separating proteins according to their isoelectric point is called isoelectric focusing. The pH gradient in the gel is formed first by subjecting a mixture of polyacrylamide amylase. So it's a small multi-charged polymers. They having many different pi values to electrophoresis. Isoelectric focusing can readily resolve proteins that differ in pi by as little as 0.01, which means that proteins differing by 1 net charge can be separated. Isoelectric focusing can be combined with SDS page to obtain very high resolution cell solution. So a single sample is first subjected to isoelectric focusing. This single lane gel is then placed horizontally on top of SDS page slab. So proteins are thus spread across the top of the polyacrylamide gel according to how far they migrated during isoelectric focusing. Then they undergo electrophoresis again in a perpendicular direction, like vertically, so to yield a two-dimensional pattern of spores. In such a gel, proteins have been separated in the horizontal directions on the base of isoelectric point and in the vertical direction on the base of mass. So like the figure shows, a protein sample is initially fractionated in one-dimensional by isoelectric focusing, and the isoelectric focusing gel is then attached to a SDS page gel, and electrophoresis is performed in the second dimension, particularly to the original separation. So proteins with the same pi are now separated on the base of mass. So as figure B shows, like protein from E. coli were separated by two-dimension gel electrophoresis, resolving more than 1,000 different proteins. So here is for today, and thank you for your attention.